On previous day, we have discussed the respiratory chain, chemiosmotic hypothesis, and inhibitor. Now today, I am discussing the uncoupler. Now what is uncoupler? Uncoupler, these are the chemical or some physiological agent that dissociate oxidation in respiratory chain from phosphorylation. Now here one chemical uncoupler, that is 2,4-dinitrophenol, and the physiological uncoupler is thermogenic. Now what is the mechanism of this uncoupler? Normally, the inner mitochondrial membrane is semi-permeable to the proton. That means the proton can translocate from inner mitochondrial membrane to the intermembranous space. And that produces the proton channel, and this proton channel helps for ATP synthesis that we know. But uncoupler, it breaks the proton channel, allowing the proton from intermembranous space into the mitochondrial matrix. So the proton gradient is broken and ATP synthesis also stopped. And thermogenin, that is another physiological uncoupler. This is actually one protein, and it is present in the hibernating animal and insect. It produces the heat. The mechanism is same, but instead of production of ATP, it produces heat. This is the physiological uncoupler or thermogenin. Now, the shuttle mechanism. No, this is the some transporter. Another is the shuttle is the next slide. The transporter. Now, the inner mitochondrial membrane, it contains several transporters. One important transporter is the ADP, ATP antiporter. This antiporter, it allows the ADP from cytosol to enter into the mitochondria for formation of ATP, or it supplies the ADP from outside. Now here, the ADP is converted to ATP, and this ATP comes from mitochondrial matrix into the cytosol through this transporter. This is very important transporter. And if this transporter is blocked, the one important drug, that is the actactyloside, it inhibits this ADP-ATP transporter. Now, important is the one shuttle mechanism, that is the mallet shuttle. What happened? The some reducing equivalent, they are produced in the cytosol or outside the mitochondria. Suppose glycolysis. It occurs in the cytosol. Here NADH is produced. So how this NADH will enter into the mitochondrial matrix or into the respiratory chain for reduction? In this case, the shuttle mechanism, it will help. The mallet aspartate shuttle, here the inner mitochondrial membrane contains some transporter. One is the mallet aspartate transport. What happens? Here, in the cytosol, the oxaloacetate is converted to mallet. This mallet, by help of enzyme mallet dehydration, this mallet entered into the mitochondrial matrix and is also converted into the oxaloacetate by same enzyme malate dehydrogenase. And if along this malate, the NADH is also entered into the mitochondrial matrix for reduction, for as reducing equilibrium. Actually, NADH is oxaloacetate and NADH is converted to malate and NAD, and this malate and NAD enter into the cytosol. Mitochondrial matrix. This malate is again converted to oxaloacetate and NAD is converted to NADH, and this NADH goes to the respiratory chain. So what happened? The oxaloacetate and NADH in the cytosol is converted to malate and NAD. This malate and NAD enter into the mitochondrial matrix, and malate is converted to oxaloacetate, and NADH is converted to NADH, and this NADH goes to the respiratory chain. Now, this malate, it cannot comes out from matrix to the cytosol. So how this subtle mechanism will be continued? One thing that in the mitochondrial matrix, the glutamate is present. In the matrix, the glutamate is present. So glutamate and this oxaloacetate is converted to the alpha ketoglutarate and aspartate. This is transamination process that will be discussed in protein metabolism. And this as alpha ketoglutarate and aspartate, it comes out to another transporter. The aspartate comes out to the malate transporter, and alpha ketoglutarate comes out to the another transporter from matrix into the cytosol. Here, alpha ketoglutarate and aspartate again converted into by another transamination mechanism, glutamate and alpha oxaloacetate. This oxaloacetate is converted to malate, and malate again enter into the mitochondrial matrix to the transporter, and continuing the shuttle mechanism. So those actually the NADH that comes from the cytosol, they enter into the mitochondrial matrix through this malate shuttle. Now, another shuttle mechanism, that is the glycerophosphate shuttle. Here, in the cytosol, the dihydroxyacetone phosphate and NADH is converted to glycerol 3-phosphate and NAD by enzyme glycerol 3-phosphate dehydrogenase. This glycerol 3-phosphate can enter into the mitochondrial matrix through one transporter. And here, one thing that in the mitochondrial matrix, this glycerol 3-phosphate again converted into dihydroxyacetone phosphate by the same enzyme glycerol 3-phosphate dehydrogenase. But one thing, remember that this glycerol 3-phosphate dehydrogenase that is present in the mitochondrial matrix, here the coenzyme is the FADH2, and this FAD and this FAD is converted to FADH2, and this FADH2 goes to the respiratory chain. So here the electron comes from the NADH, but actually in the respiratory chain, the electron goes to the FADH2, that is by complex 2. Now, what is the PO ratio or phosphorylation oxidation ratio? In the proton motive force, we have discussed that for transport of 4 electrons, per cycle, the 4 proton will be translocated to complex 1. 4 proton will be translocated to complex 3, and 2 protons will be translocated from complex 4. So what happens? In that case, the 10 protons will be translocated from different complexes into the intermembranous space. So 10 proton is to 4 electrons. So the PO ratio will be 2.5. So when the reducing equivalent will enter into the respiratory chain through complex 1, the PO ratio will, how many ATP will be synthesized per cycle? 2.5. But one thing, remember that complex 2 cannot translocate any proton. So when the reducing equivalent will enter through the complex 2 or through FADH2, the total number of proton will be translocated 6. That is the 4 from complex 3 and 2 from complex 4. And electron will be 4. So here the ATP will be 1.5. So when the complex reducing equivalent will enter to the complex 2, the ATP will be synthesized per cycle 1.5. This is all for the oxidative phosphorylation. If you go through the book, and if any query, you can contact me through WhatsApp, and I will give you all answers. Thank you.